we shall talk about the important concepts in the reproduction here now. The important points and the concepts which are likely to appear in AIMS, AFMC, JIPMAR, even EAMCET here now. Now the reproduction part here now, it is basically divided into two types, you know very well, asexual reproduction and sexual reproduction. Asexual reproduction in protozoans asexual reproduction in protozoans takes place by binary fission, multiple fission, plasmotomy, binary fission, multiple fission and plasmotomy. Now this binary fission in turn is of three types. You can say longitudinal binary fission, transverse binary fission, oblique binary fission. Longitudinal binary fission, transverse binary fission and oblique binary fission. Now the longitudinal binary fission takes place in all flagellates except Oxyrhis. There is a flagellate called Oxyrhis that reproduces by transverse binary fission. Transverse binary fission takes place in all ciliates except Verticella. Verticella, though it is a ciliate, it reproduces by longitudinal binary fission. Oblique binary fission is noticed in protozoans like Cochleodinium and Seratium. Cochleodinium and Seratium. Now the multiple fission takes place in several protozoans now. For instance, in sporozoans like Plasmodium, it is called schizogony. Sporozoans, okay, it takes place by a method called schizogony. Then it takes place even in other protozoans also, like for instance amoeba. Multiple fission can occur in encysted form or in unencysted condition. Occurs in encysted form or unencysted condition here now. Plasmotomy is the division of nuclear material without division of cytoplasm. I repeat once again here now. The other way wrong. The cytoplasmic division takes place without nuclear division. Cytoplasmic division occurs without nuclear division. The name itself indicates here now. Plasma refers to cytoplasm. Tomy refers to fission here now. So there will be division of cytoplasm, okay, without nuclear division here now. After the division of cytoplasm to form several young ones, each nucleus migrates into a small young one, a tiny young one. Later on it divides. But initially, to start with, only cytoplasm divides to form several young ones or tiny young ones. Each nucleus migrates into the tiny young one and it in turn multiplies to form several nuclei. This usually takes place in multinucleated protozoans like Pelomyxa and Opalina. Pelomyxa and Opalina here. Now we will talk about certain important concepts in this longitudinal binary fission of euglenoids. The important concepts I shall put before you, which are likely to appear in the EAMCET and all other competitive exams. Longitudinal binary fission in euglena. Euglena, it has got a terminal opening towards the anterior end, blunt end of the organism. This terminal opening is called cytostome. Cytostome is followed by a funnel shaped structure called cytopharynx. Cytopharynx opens into a spacious reservoir. Cytostome, cytopharynx and reservoir, all three of them help in egestion rather than ingestion. Then Euglena has got two flagella. There is one long flagellum here now, and there is another one is a short flagellum. The short flagellum is confined to the reservoir, it doesn't extend beyond the cytopharynx. The receptor present on the wall of the long flagellum is called paraflagellar body. The paraflagellar body is made up of a colorless pigment called lactoflavin. Which acts as a photoreceptor. Then there is another photoreceptor that lies on the wall of the reservoir here now. This is a photoreceptor present on the wall of the reservoir, almost opposite to the paraflagellar body. This is called eye spot or stigma. This eye spot or stigma is made up of red pigment granule called hematochrome. So as a matter of fact, only the paraflagellar body acts as a photoreceptor. The eye spot or stigma acts as a light filter. That is, it reduces the intensity of light that falls on the paraflagellar body. The lactoflavin present in the paraflagellar body is highly photosensitive. If high intensity light falls on the lactoflavin, it may get denatured. So it is prevented from being denatured by the red pigment called the hematochrome. Suppose you get a question here now, what are the photoreceptors of Euglena? You don't have a separate option like eye spot and paraflagellar body. Then you need to answer both of them. Suppose there is a separate option here now. Under A, suppose you have paraflagellar body. Under B, you have eye spot. Then the other two options are something else here now. Then you can answer only paraflagellar body here now. So only paraflagellar body acts as a photoreceptor. The eye spot acts as a photo filter. Suppose there is a separate option here now, you should go for paraflagellar body. If there is a combined option, you should go for the combined option, that is both of them act as photoreceptor here. So that is significance of the photoreceptors in Euglena here now. Then near reservoir, it has got a contractile vacuole surrounded by several accessory vacuoles. So this is the contractile vacuole, whose main function is excretion, main function is osmoregulation rather than excretion here now. Then it has got a distinct vesicular nucleus. Distinct vesicular nucleus is present towards the posterior end. Then it has the chromatophores. Okay, several dumbbell shaped chromatophores are present in the organism here. So these are the chromatophores here now. These are the chromatophores. Okay, so these are the chromatophores here now. The chromatophores here now synthesize food by autotrophic method here now. Then the cytoplasm of Euglena is deposited with reserve food material called paramylum. The reserve food material is called paramylum. Paramylum is said to be an animal starch as it does not give blue color with iodine here now. Then there are several myonemes that extend across the animal like this. All these are myonemes here now that extend across the animal. These myonemes are responsible for the euglenoid movement of organism here now. That is zigzag movements of the body wall brought about by contraction of pellicle and myonemes can be called metaboly or euglenoid movement. In sporozoans it is referred to as Gregorian movement here now. So with regard to the important characteristics with regard to the structure of Euglena here now. Euglena is a mixotrophic organism. During daytime it synthesizes food by autotrophic method. At night it synthesizes food by saprozoic method. So it happens to be a mixotrophic organism here. Now this Euglena during favorable conditions reproduces by longitudinal binary fission, where a groove appears within the reservoir that extends downwards, dividing the entire cytoplasm and nucleus into two identical halves. And during longitudinal binary fission, except SCP, all the other cell organelles are equally divided here now. Except SCP, there is a clue to remember this here now. Okay, in fact, SCP stands for single cell protein, but with regard to Euglena, S stands for stigma, C stands for contractile vacuole, P stands for paraflagellar body. So these three of them, all these three organelles do not divide, they disappear, once again they reappear in the young ones. Except these three organelles, all the other organelles are equally divided between the two young ones, which are called flagellulae. Longitudinal binary fission Euglena is also called symmetrogenic binary fission, as it gives identical young ones. Symmetrogenic binary fission, as it gives identical young ones. It is also called interkinetal binary fission. Interkinetal binary fission, because the fission before passing here now, it passes between the two kinetes. These two blepharoplasts are referred to as kinetes, it passes in between the two kinetes, it doesn't cut the kinetes, That's why it is called interkinetal binary fission or symmetrogenic binary fission here. The next one is the transverse binary fission in Paramecium. All ciliates, as I made a mention earlier, all ciliates except verticella reproduce by transverse binary fission here now. Now the Paramecium is commonly called slipper animalcule, which looks like sole of a slipper. Looks like sole of a slipper here now, with anterior round end, posterior tapered end. Dorsal surface is slightly convex, which is also called aboral side. Ventral surface is flat, which is also called aboral side here now. Then on the aboral side, it has a obliquely placed peristomial groove or oral groove. The peristomial groove or oral groove opens into vestibule. Vestibule in turn opens into buccal cavity. Buccal cavity. Buccal cavity opens into cytopharynx through cytostome. Opens into cytopharynx through cytostome, and the cytopharynx gives rise to food vacuoles here now. Then behind all these organelles, it has a definite aperture for the expulsion of undigested food, which is called cytopise or cytoproct, which is meant for expulsion of solid waste. It has got two contractile vacuoles, anterior contractile vacuole and posterior contractile vacuole. Each contractile vacuole is surrounded by about 6 to 10 accessory vacuoles. So anterior contractile vacuole, posterior contractile vacuole, posterior contractile vacuole. Out of these two, posterior contractile vacuole pulsates more vigorously than anterior. This question was asked many times in EAMCET, JEEE, all the other exams, under assertion reasoning. The posterior contractile vacuole pulsates more vigorously than anterior because of the proximity to the vestibule, cytopharynx and cytoplasm. Then it has got cytoplasm, which is divisible into outer ectoplasm and inner endoplasm here now. Ectoplasm is also called cortex, endoplasm is also called medulla. Below pellicle, it has got several small carrot-shaped structures are present. These carrot-shaped structures present below pellicle are called trichosis. These trichosis are meant for offense and defense. They synthesize a colloidal substance called trechin, which is responsible for offense and defense and sometimes even anchorage. Then a paramecium is a heterokaryotic organism with a macronucleus and a micronucleus. Bean-shaped macronucleus and a small micronucleus. This macronucleus is vegetative in function, controls all vegetative functions except sexual reproduction here now, and it divides by amitosis during binary fission. Then the small one is called micronucleus. The micronucleus is also called generative nucleus. It controls sexual reproduction. It divides by binary mitosis during binary fission, mitosis and meiosis during conjugation, that is sexual reproduction here now. So these are the characteristics of paramecium here now. Then paramecium during favorable conditions divides by transverse binary fission, where the two constrictions appear at the center. Those constrictions deepen and divide the paramecium into two daughter paramecia, where the anterior daughter paramecium is called prota. The anterior daughter paramecium is called proter, and the posterior one is slightly bigger in size, which is called opisthe. The posterior one is slightly bigger in size, which is called opisthe. Posterior one is slightly bigger in size, called opisthe here now. The cytopharynx does not disappear. The parent cytopharynx is transferred to the prota. Opisthe develops new cytopharynx here now. Except that all the other cell organelles are equally distributed here now. The anterior contractile vacuole of parent becomes anterior contractile vacuole of prota. The posterior contractile vacuole of parent becomes the posterior contractile vacuole of opisthe. But however, the posterior contractile vacuole of prota, the anterior contractile vacuole of opisthe are newly formed here now. The posterior contractile vacuole of prota and the anterior contractile vacuole of opisthe are newly formed here now. That it completes the transverse binary fission within 2 to 3 hours. That it reproduces nearly 4 to 5 times a day by transverse binary fission. All the descendants of a paramecium produced by several TBFs are collectively called a clone. Several young ones of a paramecia or descendants of a paramecium produced by several TBFs are collectively called a clone. In a clone, all the young ones are morphologically and physiologically, genetically, they are similar here now. Then the advantage of this particular reproduction is it multiplies very fast. However, the disadvantages are because of amitotic division of macronucleus, the young ones lose their vitality and vigor. They enter into a non-responsive phase called senescence. In order to get rid of senescence now, they reproduce by conjugation here now.